Morning, class, and welcome to our genetics course. This is our third lecture, a chromosomal lecture. This is for our May 31st class, but we'll go ahead and pre-record it today, so hopefully you'll have time to review it ahead of next week. And of course, we're going to be talking about today all about the different types of chromosomal disorders, how they come about, and how they manifest themselves phenotypically. So here we can see a nice example, trisomy 21, Down syndrome, where we have an extra copy of the 21st chromosome. So as we start moving along, when we talk about different chromosomal disorders, it's a good idea to get some of our basic terminology down. And with this, we'll be dealing with different types of chromosomes. So a metacentric chromosome, as we can see here, has the centromere right in the center of the chromosome. And then if the centromere starts to move towards one end or the other, then we'll start to have these other types. A submetacentric will have the chromosome not quite exactly at the center, but a little offset. And so now we can clearly see that shorter P arm here and that longer Q arm. Might be a little more difficult to discern the P from Q arm and the metacentric chromosome, but with special staining techniques, that shouldn't be too much of a problem. An acrocentric, as we can see, is having the centromere towards the end. So now in the acrocentric, we have a much shorter P arm and a much longer Q arm. And then last but not least, we can have this telocentric chromosome where the centromere is literally at the telomere spot. So where the telomeres would be, we actually see this centromere here. So that's a telocentric chromosome. Now, when we think about chromosomal disorders, they do come in two different varieties, and they are relatively frequent. We can see here that 1 out of every 154 births, so you imagine all the millions of births that occur in the United States each year, out of every 154.65%, that will have some type of chromosomal abnormality. Now, it is worth noting that this is actually a small percent compared to what we could have, because most or a lot of the chromosomal abnormalities will spontaneously be aborted before birth. So just a quick note that we talk about what's resulting abortion is any type of abandoning of the embryo before 20 weeks. And this could be an induced abortion or a natural abortion. In this case, when we talk about spontaneous abortion, that would be the body naturally aborting the embryo because it is not going to be a viable embryo. And so this is a nice, easy way to have a check and balance to make sure that the developing embryo would have the proper amount of DNA so that it can survive. So 40-50% will actually result in this spontaneous abortion. Now, it is worth noting that when we talk about the chromosomes disorders coming in two different types, the first type we'll look at are the aneuploidies. Aneuploidies is either having an extra chromosome or a too few chromosomes here. And so we'll see that we can either have this on the X or a Y chromosome, or we can have this on the autosomes. And as we'll see, X and Ys, they are more frequent typically because most autosomal aneuploidies will not result in a viable fetus. They will typically be lethal. You can also have structural abnormalities, maybe an inversion, a translocation, something like that. And in this case, we're going to see that we can have a few of these, which is a little more prevalent than the aneuploidies, but not a whole lot. But that gives you an idea of the different types of disorders that we'll be looking at. So, of course, when we talk about aneuploidies that come about from this non-disjunction, we can have non-disjunction at a couple of different spots along meiosis. And here we can see non-disjunction in meiosis 1, where the homologous chromosomes, they actually fail to separate during meiosis 1. Now, meiosis 2, that occurs just fine, so we can see the sister chromatids separate just fine. But with meiosis 1, one cell gets too many chromosomes, and the other cell gets none of the chromosomes. Now, when this is fertilized by a sperm cell, and this assuming the sperm cell has the proper number of chromosomes, then you can either have a trisomy, where you have 3 chromosomes, or you can have the monosomy, where you only have the 1. And of course, this could be resulting another way. You could have a sperm cell with no chromosome at that location, fertilizing an egg cell that has just the right amount. So there are, of course, other variations on this. Here we can see that we have meiosis 1 occurs just fine. So you have the separation of the homologous chromosomes. But then in meiosis 2, you have non-disjunction of these sister chromatids. So the sister chromatids fail to separate. So they both go to this cell over here, and neither of them go over to this cell over here. So once again, if you have fertilization of this non-disjunction, you can have a trisomy. And then if you have a fertilization of this other one, you can have a monosomy. And like I said, it could be the sperm cell that has the non-disjunction. It all varies. Now with that, we can take a look at some of the different autosomal trisomies that persist until birth. And like I mentioned earlier, you will have fewer of these because most of them are not viable. And so we do have a trisomy 21, Down syndrome, which is the main one that we'll be looking at in this lecture. But we also have a couple others. Trisomy 18 is Edwards syndrome, which we'll get into, and trisomy 13 is Patau syndrome. Both of these, the baby will typically live less than a year. So they are very prominent, they're very profound, and they are usually lethal. So usually these guys are going to be both lethal. Not so with the Down syndrome. Now we can have different autosomies or trisomies that are on the sex chromosomes. The sex chromosomes tend to have a little more leeway. And so we can have a trisomy of the X chromosome, triple X syndrome, so XXX. We can also have the Klinefelter syndrome where we have XXY. And then we can also have the super male syndrome, XYY. Now they call this the super male syndrome because they used to think that individuals like this, maybe in prison, had a little more aggression, a little more temper, but I think that's kind of more of a stereotype than anything. And so there's a lot of baggage that comes on with calling a super male, and they might be trying to address that as we move forward. But it's worth noting. You can also have monosomies of the sex chromosome. So monosomy X, we have Turner syndrome. Here we have 45 instead of 46. And you'll notice that we have one X, but the other spot is empty. And autosomal, and like I mentioned earlier, autosomal monosomies or trisomies, they are typically not viable because you have to have just the right amount of genetic information. You can have a few viable with the trisomies, but with monosomies, if you don't have enough, that's almost always a non-viable embryo. So once again, guys, here is our example of Down syndrome, trisomy 21, where you have 3 copies of the X chromosome. And so we're going to label that as 47. And this, of course, is a Down syndrome male because we have the XY with the extra 21st chromosome. Now, this is a nice, easy way to stain and look for trisomy 21. And we have what we can see here, fluorescence in situ hybridization analysis. And what you do is you stain the chromosomes themselves. And here we can see that we have 2 copies of chromosome 13, check, just like we should. But you'll notice that we actually have 3 copies of chromosome 21. So we can see here clearly, perhaps through amniocentesis, that we have a trisomy 21 Down syndrome embryo. There are other ways to test for this. So especially now that we're getting more advanced genetic technology, here we have the chromosomal microarray, and we can see that we can stain all the chromosomes, and you look at the fluorescence of each, and they're all typically within this normal range here. But then you see at the 21st spot, this one is clearly being overexpressed, having too much of the chromosome 21, indication of Down syndrome. And then this, kind of highlight that idea. Then we can also have whole genome sequencing. And so what we're doing is representing the sequencing of the whole genome. And once again, you can see here under normalized sequencing, all the chromosomes sit within this region. But once you get to chromosome 21, well, guess what? That's clearly having much more DNA represented in the sequence than the others. So another indication of trisomy 21. One thing that you'll notice is that under the age of 35, Down syndrome is pretty rare overall. Now, overall, we do see quite a number, so 1 in 850, so that's actually pretty common compared to your other chromosomal disorders. But most of these are going to be individuals quite often over the age of 35. And so what you'll see is that as we get over 35, you have this logarithmic expansion or increase in prevalence of Down syndrome. And so we can see a test here at amniocentesis and then another one here at birth. Now, another thing to keep in mind is that when we think about all the possibilities of a trisomy 21, most of these will also be spontaneously aborted. You're actually going to see just about a small percent, 1/5 to 1/4, 20-25% that actually survive until birth. So most of these will also be aborted during embryonic development. And of course, with Down syndrome, they have a very persistent, consistent, if you will, phenotypic expression. So we can see that we have this kind of flattened nose with these slanted palpebral fissures. You'll also notice these epicanthal folds. So the epicanthal folds, these kind of folds over the eyelids as well. You might have a protruding tongue as well. If you take a look at the eye, you can see what we call these little brush field spots around the iris. And you'll also notice that they have these small hypoplastic ears compared to other individuals. Also, you'll see a single palmar crease, so that might be kind of an interesting phenotypic expression. If you take a look at your own hand, you'll notice that you probably have a couple of those creases instead of just one. And then we also have clinodactyly and this nice wide gap between the toes. So very persistent, very consistent type of phenotypic expression that we can see here. So here we have a few patients with, or a few individuals with Down syndrome that have that typical manifestation. Now, it is interesting to know that we can have some variations on Down syndrome. And so you're going to see a small percent, about 2 to 4, are actually mosaic for trisomy 21. So if the cells early on detect that there is too much chromosome, they can do a chromosomal correction. They can either eject the other chromosome or they can turn it off like you would see it with the bar body. This doesn't happen very often, but we do see this in a few individuals, 2 to 4%, where you actually have this kind of mosaicism because some of the cells have 2 copies of the chromosome 21. Here they had a chromosomal correction, and then other individuals or other individual cell lines would have that 23rd copy. So that's kind of an interesting side note for this incident. Now, another way where we can have Down syndrome, where we have a Robertsonian translocation. What this is basically with Robertsonian translocation is you have a fusion of the 2 Q arms. So you'll notice here we have the acrocentric chromosome. So the centromeres are towards the short side here. So we have a short P arm here and then longer Q arms. And what can happen is that this Q arm can come over here and fuse together. And now we have a chromosome with 2 Q arms. And so this is what we call a translocation. This is usually between chromosome 14 and 21. The P arm, this little guy, that'll typically be lost. So we usually eject that kind of such a small piece, it's not even taken up by the cell usually. But now that you have basically 2 copies of that Q arm from chromosome 21, and so this can also result of Down syndrome. Now, interestingly enough, you could have the same number of chromosomes, right? You could have a fertilization of this cell and it would have 46 chromosomes, but because these guys are basically duplicated, you have 2 of the Q arms from 21, you're going to see, this was from 21, but you'd have another one that comes with it, fertilized. But what you'll see is that they can actually have either the right amount of chromosomes or even too few, so it may not manifest themselves in those karyotypical analysis that we've done earlier, or at least in the FISH expression. The microarrays, they would probably still show it up. So this is showing us how you would have a Robertsonian translocation. Here we have 2 copies of chromosome 14, and here we have 2 copies of chromosome 21. And like I mentioned earlier, the Q arm, so let's say that's a Q from 21, there's a Q from 14, that can come over here and fuse together. And now this chromosome, if it gets fertilized, that can definitely result. Now notice you'll still have these other Q arms that are still persistent. The P fragments, they will be lost, so that's why we don't see them in the cell lines down below. But that Q arm and then these shorter Q arms still do persist. And so what you'll notice is that if we have fertilization, and here we have couple of these gametes, you could have a normal outcome because basically you have the normal cells. These are not the ones involved in the translocation. These guys over here that were not translocated. You can, however, have what we call a normal carrier. So this normal carrier, you can see here, has the translocation, has the Q arm from chromosome 21 coming and fusing with Q arm from 14. So that's normal here. These guys, they will not themselves express Down syndrome, but they can pass it down to their offspring. And so we will see quite often that their children will always have Down syndrome, because when the egg gets fertilized, or if the sperm fertilizes an egg that's normal, you can either have a monosomy of 21 or you can have a trisomy. And monosomies won't be viable. They won't survive. But the ones that do survive, each and every one of them will have Down syndrome. I remember hearing a patient that had this incident where you had 3 children with Down syndrome that kept occurring over and over again, and that's because they were a carrier of this translocated chromosome, this Robertsonian translocation. Now you can have, of course, chromosome 21 Down syndrome, and what we can see here is that we had this non-disjunction, and then these guys are basically, if it gets fertilized by an egg, then that's going to result in a third chromosomal 21 chromosome. Now, all these guys that just have one copy of the 21, they will all be lethal. So you can have just one copy of that not the P arm, but the Q arm from 14. That's going to be lethal. The copy from the 21, because you don't have both the 14 and the 21. You just have one or the other. And in this case, you have a trisomy 14 when that gets fertilized. And once again, trisomies on most other chromosomes other than 21 will be lethal. So we can see there's only a couple of possible outcomes, but the interesting ones are the individuals that actually develop Down syndrome themselves, or these carriers that can then pass it down to the future generation. A little bit of Robertsonian translocation here. And this is a real interesting, nice kind of chromosomal theming, little fluorescence here. And we can see that translocation. So we can see that these the chromosome 14 is in yellow here, and the chromosome 21 is in blue. And here you can see that the 21 Q segment has fused to the PQ segment. So a nice illustration of how that can then come about. So there you have it, guys, a little bit about Robertsonian translocation, joining the 2 Q arms of different chromosomes together. Now, we can also have what we call partial trisomy, and so that's going to be pretty much what we see when we have just not the whole chromosome, but just a part of it coming over. All right, and we'll see that with those translocations. So there we have it, guys, just a little bit about Down syndrome. Now, the next 2 syndromes, we won't focus too much on detail because, like I said, these are much more deleterious and they are usually not survivable. Very small amount survive until birth, but a few do, so they are worth noting. Edwards syndrome is trisomy 18, so here we have 3 copies of chromosome 18, and Patau syndrome is trisomy 13, 3 copies of the 13th chromosome. With Edwards syndrome, we can see that the majority do die before birth rate, and those that do survive will have a lot of different abnormalities, heart abnormalities, kidney abnormalities, malfunctions, things like that. You'll notice some microcephaly, so a smaller head, malformed ears. They're typically in their little bit moved down and below. You'll notice a widely spaced eye, so the eyes are further apart as well, and you'll see these clenched hands and these feet as well. And here we can see Patau syndrome. With Patau syndrome, you will notice typically a midline defect. So you'll see that you may have a cleft lip, a cleft palate. The eyes may be closer together. They may actually kind of fuse together to actually form one orbit. And then you'll also see polydactyly here, where you have an extra toe perhaps as well. But like I said, Patau syndrome, Edwards syndrome, these are very lethal. They usually do not survive past the first year of life. But they are still definitely worth noting because you have a few individuals that might. All right. Now, with that said, our next group will be the structural abnormalities. So structural abnormalities can result several different ways. You can have a deletion in part of a chromosome, where you can have a duplication where you make a copy, inversion where you flip part of the chromosome over, or you can have some type of substitution or translocation. So the idea here is that this can either result in haploinsufficiency, where you do not have enough of the genetic information, or you have an overexpression of the genes that are being encoded. So you got to have just the right amount. Too little will result in bad things. Too much will result in bad things. Right. And here's a good example where we have what we call Cri-du-chat syndrome, and this is named for the cat cry that is often associated with the babies from this individual. And this is a deletion at the 5p. So here we see a 5p chromosome, and we'll notice that the end here has been deleted. And this is what we call cry-du-cat or cry-du-chat syndrome. And that's due to the typical cat-like cry of the affected children. So this could be either a terminal deletion or an interstitial deletion somewhere in the middle of that. And depending on where the deletion occurs, the phenotypes will vary. So that's going to be a part of this as well, depending on how the where the deletion occurs, the phenotypes will be a little bit different, as we'll see. And this is highly variable at the breakpoints, means variable expression. So what we say here is that this is going to have different symptoms due to haploinsufficiency. And we can see here, if you have just the tip of the P arm deleted, well, you may have an individual with speech delay. As you move further down, you're going to have maybe the cat-like cry. And then as you move further down, that can result in microcephaly, severe mental retardation, also facial dimorphism, either in the childhood. So you'll notice here is the childhood, and as you move further down, it persists to the adulthood as well. So as we can see here, moving further and further down on the P arm here will result in a variable expression of this disorder or this syndrome. And so what you'll notice also, symptoms are due to the haploinsufficiency, and vast majority of these do not have some type of hereditary thing, where we see that most of these are due to de novo mutation. So this is the first individual in that family history to occur with that syndrome. So Cry-du-Chat syndrome. A couple things that we can notice with Cry-du-Chat syndrome: hypertelorism, and what that means is that you have this widening of the eyes. There's a widened space between the two eyes. The epicanthal folds, we saw this with Down syndrome, and we see it here where you see that fold over the eyelid. And you'll also notice we have retrognathia, and what that is that kind of receding chin, that mandible, if you would, on that front. It's a little more flattened, if you would. So Cry-du-Chat syndrome, just to give you guys an idea of what to look for. Here's an individual that we can see as they progress through the years. So we start at 2 years old, 4 years old, 9 years old, and 12 years old, just to give you kind of a developmental picture of Cry-du-Cat or chat. All right. Now, another chromosomal disorder that we can have can come about through translocation. So translocation, as we can see here, will occur when two different chromosomes, let's say this could occur between any two chromosomes, but let's say we have chromosomal 11 here and chromosomal 14 here. Well, when they swap genes, you might have some type of translocation. Now, this will typically occur whenever the translocated chromosomes form this, what I call a quadrivalent. So under normal circumstances, as the homologous chromosomes pair together, they would form separate bivalents. So maybe chromosome 11 here is forming its bivalent, whereas chromosome 14 is forming its bivalent. But as they swap genetic information, as we can see here through this translocation, well, now chromosome 11 have a piece of it over here on chromosomal 14 and a piece of chromosome 14 over here on 11. And so they're going to result in this very unique, very interesting type of alignment, that quadrivalent with that translocation. So this is just showing you kind of how we form these quadrivalents. And so let's say we have our two chromosomes. And like I said, this could be any two chromosomes, but for our sake, we'll just say chromosome 11 and chromosome 14 here. And we can see that we have these different regions on each of the chromosomes. And in order to actually align for meiosis, each of these guys will have to align their different segments. So when you get this translocation, let's say we have that Q arm from 14 coming over to 11 here, and then the Q arm from 11 going over 14 there. Well, now you're going to have that quadrivalent forming, and we can see here it's required in order to get that proper alignment of each of these segments. Now, depending on how the translocation occurs, this can either form a balanced or unbalanced gametes, depending on which segments had actually been translocated, which ones had been moved over. And so here we can see a couple of examples of translocations, whether it's balanced or unbalanced. And in this case, you can have separation, for example, of the quadrivalent along that horizontal axis. And so these guys could come over one cell and this one the other cell. And these will both be unbalanced because all the genetic information is going to one side, not the other. So these will typically not be viable, or they could result in some type of disorder if they are. And another way that we can have it is that we have separation along the vertical axis. So in these guys, we're going to separate here in this way. And once again, these guys will be unbalanced and resulting in some type of typically a non-viable embryo, if you would. But you could have separation of the quadrivalents along the diagonal here. So these guys would come over to one cell, and then these guys would come over to the other cell as they start to separate. And what you'll notice is, okay, that's normal because you'd have that normal separation like you usually would, but you could also have a balanced separation of the chromosomes, or pretty much you get the chromosome 11 and chromosome 14, the parts that have been translocated, they're complementing each other. But now you'll notice that these chromosomes are completely abnormal, and so as they get further passed down, that can then result in some chromosomal issues, some chromosomal disorders. So a little bit about translocations and how they occur. And like I said, a lot of these guys will be unbalanced. They won't be viable. But if you get this separation along the diagonal here, then you can have a nice balanced individual where they have enough genetic information, but now when they have children of their own, that's going to lead to the different chromosomal problems that we'll be looking into. So here we can see an example of balanced translocation, and you're going to see that this occurs in about 1 in 2,000 newborns. So generally no obvious phenotypic defect because most of the genes are there. They might have a little bit if the translocation occurs and breaks a gene, but usually that gene is expressed elsewhere, so it's kind of being made up for. So very typically you won't see no general obvious phenotypic defect unless you get a disruption of an important gene, which usually doesn't happen. But all right. Now, let's suppose we have a balanced translocation. So here we can see that we have a translocation 1421, and you're going to see when they have their own children, that balanced translocation then can result in other disorders. In this case, you can have a trisomy 21 Down syndrome, right? And that's going to be if that balanced translocation gets fertilized and then that individual will have too many. Of course, you could also have formation normal gametes because you could have the balanced translocation if it's fertilized by a normal gamete, that's going to cause trisomy 14, which of course is not viable, and monosomal 14, which is not viable either. But these individuals could also be a balanced carrier, so if it's getting passed down, and then these guys, individuals, as they are carriers of that gene, that translocation, they could then pass down the disorder themselves. So you'll have an unbalanced gamete in F1, and then potential for an imbalance in F2, depending on how the chromosomes are passed down. All right. Our next group of disorders are those that are involved in inversions. And so inversions are when we are flipping segments of the chromosome, and we classify them in two different ways. They can be pericentric, which means they're occurring around the centromere. So you'll notice over here we have A, B, C, D, and E. Well, A and B are above the centromere. C and D are now below it. If you have inversion now, you have the C coming above the centromere. Now it's on the short P arm, and the B is now down on the Q arm. So that's going to be around the centromere. A pericentric inversion, that occurs on one side of the centromere or the other. So notice in this case, A, B, C, and D. B, C, and D are both on this side of the centromere, and then they can get inverted as well. And that would be classified as a pericentric inversion. So we'll have some consequences occurring because of these inversions, depending on where they occur at. So one thing you'll notice is that because these guys are inverted, in order to properly separate, you'll notice that they'll actually have to do this kind of loop to align the D's and the B's together, right? Because these guys are not going to be able to align straight on. So you're going to have this kind of looping effect here, and then you can have that crossing over effect, if you would. And when that happens, then you can either have a balanced chromosome. So you'll see here we have all the genetic information, A, B, C, and D, just like we had before. And A, C, B, and D, all the genes are there. They're just in a different order. But you could also have what we call it dicentric. And dicentric is where we see that we have two centromeres on one chromosome. So in this case, both centromeres have joined on to one chromosome. So there's one centromere and the others. That's going to be a dicentric. And then we can also have an acentric. Acentric is having no centromeres, and that will also be inviable. So either having too many centromeres or having too few will definitely result in that. But like I said, you can get that inversion, and then you can have that balanced, if you would, and that's going to be a nice little mutation of that chromosome. Now we talk about pericentric. So pericentric, we're going to be going across here. Here we have A, B, C, and D. And now we're going to be going across that centromere here. And what we can see here is, in order to align these guys, to align B and B over here, and C and C over here, we will once have to have that kind of inversion, this kind of looping, if you would. And so now what we'll see here is that we get this recombination, and the recombination can either be balanced. So once again, we have A, B, C, and D, just like you would in the normal. But we could also have this A, C, B, and D, where B is now on the other side of the centromere. It's now on that P arm there, or and I guess over here in the P arm. But now you can see we still have the complete set of genetic information. So that's going to be a balanced inversion. But once again, guys, we could have an unbalanced, because now this chromosome has 2 copies of the A gene, and this chromosome over here has 2 copies of the D gene. So the chromosomes have basically joined onto the different, or the genes have joined on, I'm sorry, I should say the loci. I keep saying genes. The loci are joined on fused onto the same chromosome. So once again, that can either be balanced or unbalanced with that recombination event after you get the inversion. All right. Our next genetic disorder is a very interesting one, and actually a couple of them, but they both result from what we call genetic imprinting. And genetic imprinting is a very interesting trait, which we'll talk about here in a little bit. But this is going to result in either Prader-Willi syndrome or Angelman syndrome. And what we can see here, in this case, a nice little karyotype where we have a 5 megabyte deletion in a band that you would typically not be able to see by normal karyotyping. But these micro deletions, a tiny deletion on a small spot, we can pick up. And we're going to see this occur with both Prader-Willi's and Angelman syndrome. Now, when we talk about these micro deletions, they're both occurring on chromosomal 15, and this chromosome will actually be responsible for this genetic imprinting. Now, when we talk about imprinting, what we're saying is that your chromosomes, as they are passed down to the next generation, parts of them are marked as whether they're coming from the mother or the father. So if you guys remember a little bit about epigenetics, we can actually have hypermethylation. So we can see here, for example, the paternal chromosome that came from this from the male is marked at this location with hypermethylation, as is the maternal chromosome. And so that way, when they come together, these areas that are hypermethylated, they won't actually be expressed, because if this area here is hyperexpressed, or if it's methylated here, that's going to turn that gene off, and then the maternal chromosome will actually cover for that. Likewise, if the maternal area over here is hypermethylated, this chromosome will be turned off here, that'll be shut down, and so this one will need to be expressed. So you're going to get an alternate expression on the chromosomes depending on which one is hypermethylated. Hypermethylated basically means it is being turned off, if you would, in one sex, and then the other chromosome from the other sex would have to turn it on. Now, it is worth noting that after you get fertilization and then the gametes form, that these hypermethylations, they do get erased, so that the next generation, they will have their own genetic imprinting as they pass down their chromosomes, whether you're a male or a female. But it's a very interesting type of marking of the chromosome that has some very unique consequences. So, for example, just as a reminder, so here we have in the male, his chromosomes are marked. His chromosome here is marked here, and that's coming from his mother. And then you'll have that spermatogenesis. And then, of course, you're going to have this chromosome marked coming from the male. So that's going to be in the sperm, and that's going to fertilize this egg that came from the mother. And like I mentioned, once these guys fertilize an egg, that will then make a new generation, and then they will erase and then remark the chromosomes for the future gametes. So anyway, so after fertilization, both male and female embryos have one copy of the chromosome carrying a paternal imprint and one copy that carries a maternal imprint. So this one is imprinted. And when I say it's imprinted, that means it's hypermethylated. It's turned off so that the paternal chromosome will take over. And like I said over here, when that's hypermethylated, when it's imprinted, that will be turned off. So the maternal chromosome will have to take over. And so Notice that during oogenesis and spermatogenesis, the imprints are erased by removal of the markers, and then you'll get those new imprints with the sex of the parent established as they're passed down. All right, this is where things get really interesting with Prader-Willis and Engelmann syndrome. And what you'll notice here is we have a paternal region on this chromosome 15, and we also have a maternal region over here. And what you'll notice, like I mentioned before, is that if you have a deletion along these areas, so you can see here a deletion, that's going to result in either Prader-Willi syndrome or Engelmann syndrome, depending on where the deletion is and depending on whether it's being, the disorder is going to affect the paternal region that's imprinted or the maternal region that's imprinted. So let's talk about the paternal region first. And this is going to be dealing here with this expression that's only passed down only from the paternal chromosome. So this expressed region over here is on the paternal chromosome, which means on the maternal chromosome, it will be imprinted. It will be turned off. Likewise, over here, this area is only expressed on the maternal chromosome. So that means on the male chromosome, it will be turned off. So hopefully that gives you a little bit of an expression here, but this area here, Prader-Willis, it's only expressed on the paternal chromosome. It's actually imprinted on the maternal chromosome. And then over here, Engelmann syndrome, that's actually expressed on the maternal chromosome where it's imprinted or turned off on its homologous pair of the paternal chromosome. So what we can see here, we get these different regions that can result in these deletions. And what happens is when they're deleted, those genes that are normally expressed, they won't be expressed anymore. And so that's going to be basically preventing the other, or that's going to be causing the other chromosome to actually be expressed. Normally the other chromosome should be imprinted, it should be suppressed, but when we have a failure to suppress it, that chromosome then that's normally imprinted will start to get expressed, and it's that expression that will result in these different these different manifestations of Prader-Willis and Engelmann. So that might be a little bit confusing, I know, but hopefully this here will help clear it up. So let's say we have this chromosome 15 normally containing a genomically imprinted, and these genes are only expressed on the paternal and the maternal, depending on which one you're talking about. Now, like I said, on the opposing chromosome, on the opposite chromosome, it will be hypermethylated. It will be turned off. It will be imprinted, and so that it will be shut off. So what happens then if you get a deletion in the expressed area, then you fail to imprint. You basically disinhibit that inhibited area. Yeah. All right. So let's talk first about Prader-Willis and see if this clears things up. So here we have a paternal chromosome, and here we have a maternal chromosome. And notice on the paternal chromosome, we have that Prader-Willis syndrome or region here, and here it is on the maternal chromosome. This region on the maternal chromosome should normally be imprinted. It should be turned off. So as long as this guy is doing its job properly on the paternal chromosome, this will not be expressed. However, what you will notice is if you get a microdeletion along this area at any point, that will suppress that gene on the paternal chromosome. And because this is being suppressed, this will start to be transcribed and it will be turned on. And so when we turn that on, then we're going to start to see that these results of Prader-Willis. So it's a failure to imprint that region that we were supposed to imprint on the maternal chromosome. So you're going to see this as very kind of rare, 1 in 15,000 births, but that's a microdeletion on the paternal chromosome, and that basically fails to imprint the maternal chromosome like it's supposed to be. However, they figured this one out is definitely an interesting topic to go into. So the idea here is that if you have no Prader-Willi syndrome gene expression, then that will result in Prader-Willi syndrome. So with the expression. So what do we see with Prader-Willi syndrome? Well, you're going to see it starts at infancy. You're going to see, interestingly enough, during infancy, you have difficulty feeding the children. The child may have a suppressed appetite. But after infancy and childhood, you're going to have this very large appetite, just always hungry, hyperphagia, and it's going to result in obesity, so never satisfied. You'll also see hypotonia, rather, this weakness of the muscle, and some cognitive impairment. Not as severe as Engelmann syndrome, but we'll still see some of it. And a shorter stature. And then you'll also see some different dimorphisms. You might see smaller hands, smaller feet, as well with Prader-Willi syndrome. So just think about Prader-Willi's, always hungry, and a little bit of cognitive impairment and a hypotonic muscle tone. All right. Now let's talk Engelmann syndrome. Engelmann syndrome is actually going to result from a microdeletion on the maternal chromosome, not the paternal chromosome, at the similar location. So we're still on chromosome 15, still the same setup. Once again, we have our paternal chromosome up here, maternal chromosome down here. Now in this case, with the Engelmann syndrome, it's on the paternal region that should be imprinted. This area here should be turned off. As long as this chromosome down here is working just fine, or this gene here is working, this paternal chromosome will be turned off. It will be imprinted. Now with Engelmann's, if you have a microdeletion at any point along this area here, that will fail to imprint the paternal chromosome. And when we fail to imprint this paternal chromosome, this is when we start to see Engelmann syndrome. So here we have no Engelmann syndrome gene expression. And if you don't have that gene expression here, this will get turned on and it will cause the result of that Engelmann's. Now with Engelmann syndrome, you're going to see some major phenotypical features. This is often what we call the laughing puppet syndrome because you think about very much laughing and these kind of mechanical-like movements. Microcephaly is one of them, a shorter stature, often involved in seizures, minimal speech, and we're also going to have cognitive much more delay than we would see in Prader-Willi's. Not much speech at all. Frequent smiling, frequent laughing, like I mentioned, the laughing puppet, if you would. And of course, some developmental delays with severe intellectual disabilities. And you'll also notice widely spaced teeth as another phenotypic of Engelmann syndrome. So there you have it. Hopefully that wasn't too confusing going through Prader-Willi's and Engelmann's. There's two definitely very unique types of chromosomal abnormalities that we're dealing with here. So what we can see here is that a lot of these are going to be dealing with chromosome 15. And here we can see the deletions, the microdeletions are occurring on the Q arm, somewhere between typically 11 and 13 of that microdeletion. And what we'll see here is on the paternal chromosome, 70% giving us Prader-Willi syndrome, 70% for the Engelmann syndrome. That's depending on whether it's on the paternal side or the maternal side. Now, you could also have another way that this could come up at a fewer, 30% rather, is what we call uniparental disomy. Uniparental disomy, where we can see that's only going to be primarily in Prader-Willi syndrome, and you're going to see that's occurring in 30%, 20%, much fewer in Engelmann syndrome. All right, so what is uniparental disomy? Well, let's take a look. So here we have our normal chromosome, and let's suppose we get this non-disjunction during meiosis 1. And with that non-disjunction, then we get 2 copies of chromosome 15 over here and no copies over here. And of course, we're dealing with 15 since this is Engelmann and Prader, just for our example. Now, let's suppose we have our normal meiosis, and remember we have the non-disjunction, and so here we have 2 copies of chromosome 15, and here we have a third one coming in. That'll give us the trisomy that you might expect. Now, because this parent is coming from the paternal and the maternal, so that was the maternal chromosome 15, that's the paternal, and that's the sperm, that's going to give us what we call heterodisomy, or these are all different. Now, if you remembered early on in development, if a cell detects that it has 3 chromosomes, it can do what we call a chromosomal correction. And that can eject part of that chromosome. And it's just random. It can eject one of the 3 chromosomes at random. And in this case, it just happens to be that sperm. But now we have trisomy rescue, where we reestablish our diploidy or diploidy. And notice that we now have 2 copies in that fertilized egg, but these 2 copies are both from the egg cell, the egg cell. And so that's what we would call a heterodisomy. And if that happened to have that microdeletion area on chromosome 15, that would then express the condition. Now, another way we can have this is what we call isodisomy. And isodisomy, what we'll see here, is meiosis 1 goes just fine. Meiosis 1 is good to go. But now in meiosis 2, we have failures to separate the sister chromatids. And when this happens, notice the sister chromatids do not separate. They both go to this cell. And these are the identical. So isodisomy, because you have identical chromosomes over here. Now, in this case, this could be fertilized by a sperm with no chromosome. So notice that chromosome 15 is missing from that sperm. It just happens to have none at that one location. And if that happens, we would still have a fertilized egg that would still give us the microdeletion because we have 2 copies from the maternal chromosome. So isodisomy and heterodisomy, just to show us a couple of different examples of disomies in this condition. So like I said earlier, disomy is not very common in Angelman syndrome. It's only 7%, but you do see it relatively frequently, relatively speaking, in Prader-Willi syndrome, where you get that uniparental disomy. 30% of your microdeletions will result from that, getting 2 copies of those chromosomes. All right. Now, with that said, let's talk sex chromosome disorders. So sex chromosome disorders, they are more lenient with their lethality because we have ways to get around that. So when we talk about this, there are a few different types of sex chromosomal or I should say, first of all, there are 4 stages of sex determination, and all 4 of these have to go properly in order to get the result that they get the proper sex. So first and foremost, you have to have chromosomal sex determination. So it's going to determine whether you have an X or a Y. So that's going to be a male. That's going to be female. But that's not sufficient to determine male or female. After that, we will then have to have determination of the gonads. We'll have to determine whether we have the ovaries, the testes, and of course the differentiation of the genitalia. And you'll have to determine the sex hormones, whether you're secreting estrogen or testosterone. And finally, we have to determine sex by the development of secondary sex characteristics. So guys, as you can imagine, there are several steps along the way, and all of these steps are each influenced and controlled and determined by genetic factors. And each of these steps can go awry. So what we'll do here is take a look at some of these different disorders that occur with sex chromosomes. So let's start here with the Y chromosome, and you'll notice the Y chromosome is, of course, only has a small area that's actually homologous. This is what we call the pseudo-autosomal region. So if you guys remember the short P arm, we have up here our sex determining region on the Y chromosome, which we'll label the SRY. And then we'll also have with it the testes determining factor, TDF, on the Y chromosome as well. But those areas are going to be the ones that are basically similar to what we would see on the X chromosome. Now you'll also notice that we have multiple regions associated with disorders of sexual development. And we'll talk about these different DSDs, these disorders of sexual development, but depending on which region of the chromosome that gets affected, the disorder would vary in its phenotypic manifestation, as you can imagine. All right, to understand this, we should first understand how sex is determined to begin with. And it's worth noting that the default sex is, of course, female. So in the embryo, unless instructed otherwise, will develop into females. It is through the sex determining region, the testes determining factor, a variety of other factors that will convert that default female sex determination into the male. Now, to understand this, we're going to start with a primordial gonad, and this is ambipotent. So this can either become the testis or the ovary, depending on how it's affected. And you'll notice that we have a couple of tubes that are going to be connected or here associated with it that will determine or partially determine how this develops. Here we have what we call paramesonephric duct. The old name for this was the Mullerian duct. And then over here, we also have a mesonephric duct, and the old name was the Wolffian duct. So, two different ducts to deal with, the paramesonephric or the mesonephric duct will influence the formation of this gonad, this ambipotent gonad. Now, if we think about what's going on with sex determination, at 6 weeks in utero, as long as we have the Y chromosome, that's present, As long as we have a functioning SRY, the sex determining region on the Y chromosome present, check. As long as we have testes determining factor, check. Then we will get development of a testis. Now, assuming all of that occurs properly and you develop the testis, then we need secretion of testosterone, and we need another hormone called anti-Mullerian hormone, check. As long as those two are secreted properly, then we will get formation of the male genitalia. So all of these steps have to occur. You have to have the Y chromosome, functioning SRY, TDF. You have to develop the testis. Then you have to have enough testosterone and secrete the anti-Mullerian hormone to get your male genitalia formation. And all of this starts at 6 weeks. A lot going on there, right? Now, let's suppose this doesn't happen. One of these steps does not occur. You don't get the Y chromosome. Maybe the Y chromosome doesn't have SRY. Maybe it's not releasing TDF. Maybe you're not getting AMH. Then, at 8 weeks in utero, we will start to have formation of the ovaries. So what will happen is that ambipotent gonad will start to develop into an ovary. That will start to secrete estrogen. That will then develop the paramesonephric duct. And after all of that occurs at 8 weeks, then you'll start to have formation of the female genitalia. So making sure, hopefully we start to understand what it takes to become a male and what it takes to become a female early on in development. All right. So now we can start thinking about how these are going to be infected by your genes or affected by your genes. And of course, one region is what we can think of is when we have recombination outside the pseudo-autosomal region. So you guys remember the pseudo-autosomal region where we have the X and the Y. There's also another one on the other end down here, but we won't need to look at this for our purposes. We'll just focus on the P arm in this case. Whoops. And let's suppose, for example, we have a crossing over event. And notice in this crossing over event, here's the Y chromosome with its sex determining region and a testes determining factor, and it happens to cross over with the X chromosome. And now the X chromosome, it has, guess what, the sex determining region on it, the testes determining factor, and that can give us, because it has both of these, a male that has 2 X chromosomes, an XX male, and that's because it has both the SRY and the TDF on that X chromosome. Or if this Y chromosome happens to fertilize an egg cell, well, because it's lacking, because it does not have the SRY, because that's missing, because the TDF is missing, it will actually develop into a female. So in this case, you would have a XY female. So just to give you one example of how this can occur. And so, oh yeah, I forgot I had my little illustration in there. Kind of zoom in on what's going on. SRY, TDF now coming over to the X chromosome and leaving the Y chromosome to give us those phenotypic manifestations. So in this case, we would consider this 46XX male, 46 chromosomes, but it's a male. And then over here, we're going to have 46XY female. So very interesting phenotypes that can result from this. All right. So like I mentioned, there are multiple possibilities for disorders of sexual development, because if any of those pathways gets disrupted, then that can result in a disorder. So you can have anything from complete gonadal dysgenesis to a mixed gonadal dysgenesis or ambiguous genitalia. So you can either have males that have one testis, one ovary. You can have a gonad that's half testis, half ovary. All sorts of things along this. So you can have external genitalia that are female, internal genitalia that are male. It goes the whole spectrum pretty much. All right. So let's talk first about disorders associated with the 46XY karyotype. So one thing that you can have is an XY complete gonadal dysgenesis. And so what we'll see here then with gonadal dysgenesis is that you're going to have either a mutation or a deletion of the SRY. And because of that, you can have SRY mutation, you have duplication in some areas. These are more complicated. We won't worry about that. But what it will do is it'll basically, fail to form the testes like it normally should. So a lot of these guys will have a normal SRY region, And that's because there are several different genes, as we're learning more and more about, not just SRY, that are responsible for this pathway. So a mutation that affects any of those genes can definitely affect complete gonadal dysgenesis. Other things that we can think about further down the line, we could have what we call androgen insensitivity syndrome. So maybe you make the testes, maybe you make the androgens, but the receptors for the androgens may be defective. And if the receptors for androgens do not work properly, then you would not be responsive to androgen. And this, of course, comes in a couple of varieties here. You can have a complete androgen insensitivity syndrome. So you don't respond to androgens at all. Or you could have a partial androgen insensitivity syndrome where you respond to it a little bit, but not fully like you should. So here's a good example of androgen insensitivity syndrome. And this is what we call guavedoses, if I can pronounce that right, guavedoses. Literally translates into penis at 12. And this is actually common in an area, I think it's the Dominican Republic in this case. And what you'll notice is that these children begin to develop as girls, as females, and then all of a sudden at age 12, they turn into males. So imagine going your entire childhood life as a girl, and then all of a sudden, right when you start to hit puberty, get to 12 years old, guess what? You start to develop a penis. So I'm sure that as if development isn't complicated enough, guavedoses could definitely complicate it exponentially more. But this is something that we see in, I think, Dominican Republic, if I recall it correctly. All right. Now, there are a couple things that we can think about. One of these, we can have what we call androgen activity syndrome, where we fail to either activate the androgen or when it does become activated, doesn't quite work properly. And you can see there are several steps along the way in order to make the androgens, and this is all derived from cholesterol. So, for example, we can at this step convert testosterone into DHT, dihydrogen testosterone. And what can happen then, like I mentioned earlier, is if the protein doesn't work for the receptor, if that's defective, that can result in androgen insensitivity. So this is usually due to a mutation on the gene that encodes the androgen receptor. So even though you typically make enough androgens, the receptor won't work properly, and you will become insensitive to the androgens that are produced. So another way that this could happen is very rare, but you could also have the failure to make DHT to begin with. So there's an enzyme here called 5-alpha reductase, and that's responsible for converting the testosterone into DHT. And we can see that's in the very last step. Well, if you have a defect in that enzyme, then you won't be able to make the DHT. You'll make the testosterone, but not the dihydrogen testosterone. That's the active version, by the way, of the testosterone. So, yeah. Now, so in this case, you get normal testosterone production, check, but it cannot be converted into that DHT. And now what we'll see here is that the patient will actually have external genitalia as it appears as a female, but the internal urogenital organs are male. So they'll have the male vas deferens, perhaps, seminal vesicles and whatnot. So what you'll notice here is that they can have elevated testosterone and they will have a low DH. So we'll often look at the what we call testosterone to DHT ratio, the ratio between those two, in some type of disorder like this. All right. So now that we take a look at a couple of these 46XYs, let's take a look at disorders associated with XX karyotypes. And what you'll notice in this case, they will typically have a translocated SRY and start to develop as males. And so we can have what we call XX testicular disorder of sexual development. And that's kind of what we see over here, where you get that translocation of the SRY gene. You will notice there are a couple of other possibilities. These are other genes along the pathway that can also result in the same phenotype response. So you will notice that about 15 and 20% have no SRY, and they have ambiguous genitalia, typically from these other results, SOX9 duplications and whatnot. And of course, you can have evidence of male sexual development. So cryptorchidism, hypospadacia as well. And we'll take a look, I think, at hypospadacia, if I remember right here in a second. So One thing you will notice is that in some individuals with 46XX, some will actually have evidence of both testes and ovaries, and there's a couple ways this can happen. One thing you could have if the tissue itself could have what we call a ovotestis. So this tissue has both the characteristics of an ovary and a testis, and so it forms what we call the ovotestis. So let's say, for example, you have an SRY translocation, and then you could have this kind of development of the ovotestical disorder, and then you could have some type of formation of the ovotestis. Now, one thing you'll notice is congenital adrenal hyperplasia. If you guys remember, adrenal glands are also secreting testosterone as well, and so that can also result in this condition. Another way to get the testosterone with this congenital adrenal hyperplasia. And then you're going to get that XX virilization. So, why can we tolerate sex chromosome aneuploidies, right? Why autosomals are mostly lethal, but with sex chromosomes, we tend to have more leeway. And hopefully, as you start thinking about what's the difference between sex chromosomes and autosomes, what makes them special, well, hopefully you guys are thinking about your Lion hypothesis or Lion theory about X inactivation, right? Where we have those bar bodies where, because males have one X chromosome and females have two, females will typically suppress or inactivate one of the X chromosomes at random. So, and this will allow for more leeway with the X chromosome aneuploidies. So aneuploidies for the X chromosome is among the most common cytogenetic abnormalities. So very, very common. And you're going to see it's a dosage composition dependent. X inactivation equalizes the expression of the X-linked genes between the two sexes. So, for example, here you can see two different individual or two different cell lines. Rather, it could be the same individual, and in one cell line, the paternal chromosome becomes inactivated. It becomes a bar body, so that only the maternal X chromosomes persist. And this other cell, the maternal chromosome is inactivated, and so only the paternal chromosomes are expressed. And that's where we get that kind of mosaicism that you guys might be familiar with, calico cat, for example, where we see those different patches of colors along the fur. So a little bit, there's my pen. So a couple things to keep in mind. You'll notice that when we talk about X inactivation, it'll be -1 of the normal X chromosomes, right? So it'll be one less. So for example, here is one inactivated X chromosome. That's a bar body. And because we have a bar body there, hopefully that will tell us that that is actually a female. A male would not have a bar body because that X chromosome would not be inactivated. They need that one X chromosome. So by having that bar body there, that's a good way to look at it. Here over here, you'll notice here is 3 inactive X chromosomes. So it's these 3 bar bodies, 1, 2, 3. And then of course the other X chromosome is not inactivated. And so it is not appearing as a bar body. And so look at this, 49XXXX male. So you can get 4 copies, 5 copies, maybe even 6 copies of this X chromosome and still have a viable offspring. So a nice little fish for those histone varieties of those different number of X chromosomes. And of course, that'll have an impact on the X-linked gene disorders. Supposed to have an E there, but there you go. All right. Now, one thing to keep in mind, guys, in order to inactivate the X chromosome, we need the X inactivation center. So there's an actual center here, a part of the gene that is responsible for inactivating the X chromosome. And this is critical for that X inactivation. And so we can see here, when we have that gene, then you get inactivation of that X. And now you just have this monoallelic expression of the opposing X chromosome. So you do need 2 X chromosomes for ovarian maintenance. You have to have both of them, but only one of the X chromosomes will actually be activated. So you got to have 2. You can't have just one, but only one will be actually in play because the other one will actually be inactivated. All right. So let's talk about some of these major sex chromosomal abnormalities then. Of course, we have Klinefelter syndrome. We see Klinefelter syndrome here, XXY. So we have 2 X chromosomes and a Y. And then you can also have trisomy syndrome, which we see down here, triple X, so 3 X chromosomes. And then we can also have XYY, where we have 2 copies of the Y chromosome. remember the super male. And then you can also have Turner syndrome. Turner syndrome is the XO, so you get just one copy of the X. You could have an X chromosome with a partial deletion of the other chromosome, or you could have where we have this inactivation of the deletion. So a few different ways you can have Turner syndrome, but basically XO. And then, of course, you can have some Y chromosomal rearrangements, some Y deletions, if you would. We won't really get into Y deletions, but... All right, let's talk about Klinefelter syndrome. So Klinefelter syndrome, or XXY, you get this 1 out of 1,000 live male births. And you'll see these patients typically are tall and thin and have these relatively long legs. And they appear typically normal until puberty. And you're going to get this hypogonadism start to become more and more obvious. So you'll have maybe a smaller penis, for example, or smaller testes. And you'll notice that the breast, gynecomastia will typically be enlarged in males. So that's just referring to having in males these enlarged breasts. So a little bit about Klinefelter syndrome. Turner syndrome. Turner syndrome is our XO syndrome, where you have 45 copies of the chromosomes because you're missing either an X or a Y over there. And you're going to see about most of these are not viable. Once again, you have to have enough chromosomes. 98% will spontaneously abort. The ones that do, you will see about 1 in 2,500 to 1 in 4,000 females, and they do have some characteristics that are phenotypically visible. Typically shorter in stature, so if you notice our growth scale here, measuring the height, here in the dotted lines, that's normal, and then down here, this would be Turner syndrome. And you can see they fall below the growth chart. Also notice a webbed neck, so you're going to have that kind of webbed neck appearance, along with lipidemia, all part of Turner syndrome. Here we can see the XXX syndrome, triple X, 47 triple X, and this will typically cause no unusual physical characteristics. Most females will have normal sexual development, and they are able to conceive children typically just fine. So you might have an unusually tall female, but some of these are thought to have a small increased risk, but not necessarily a consequence, just maybe a risk of developmental delays, disabilities of speech and language. But that I'm not for sure how well established that is. So it's a increased risk, if anything. So this kind of shows us a nice overview of the different sexual disorders. So here we have Klinefelter syndrome, or XYY. Notice XYY occurs in about 1,000 males. Tall but otherwise appearance typical. They say we're not for sure exactly how accurate the low IQ is, but you might have a little bit of language delay, reading difficulties, and sexual development is apparently normal. Trisomy, or triple X syndrome, which we covered, also 1 in 1,000 female births. And then here we have a few different things, maybe a little bit of hypotonia, a little bit loss of muscle tone, but maybe not manifest itself, but taller than average is one that we typically see. And then here we have our XY Turner syndrome. So there you have it, guys. A few different sex chromosomal abnormalities. Now when we talk about the overall incidence of sex chromosomal abnormalities, well, this will occur in about 1 in every 300 males will develop some overall sex chromosomal abnormality. Compared to females, that's about 1 in 650. So pretty prevalent when you consider 1 in 300 males, 0.3%, 650 females. So when we consider both of those together, you can expect to see some of these probably in the clinics as well. Well, there we have it, guys. Our chromosomal disorder lecture. You all have yourselves a good week, and I will see you back in class following next week. Take care.